So what does DNA look like? DNA, you should know the word nucleotide, right? That's the monomer of DNA. So we're going to draw one nucleotide right now. Draw this with me. Sharpen pencil so you can erase. Clean sheet of notebook paper. This is DNA structure. If you want to title it, I'm gonna leave the title off because I'm gonna need the space. But the double helix structure, the structure of DNA. We are gonna have a phosphate. The phosphate, which is going to look in this model like a circle, is going to kind of come off. There's going to be a bond that kind of looks a little bit like this. And this is going to be attached to a five-sided molecule that kind of looks like a house tilted a little bit. And then this is going to be attached to, we're going to make this a rectangular piece. This is going to be the deoxyribose sugar. That's the house shape. Deoxyribose. That's the sugar where DNA gets its name from. And then this is a nitrogen base, right? Nitrogen base. And now, I don't want to get too technical here, but the sugar, notice, has 5 points to it. This is a carbon here. 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 This point right here is actually where an oxygen is, so there's no carbon here. The 5th carbon is on a little molecule, a little, I should say, little functional group that sticks off right there. So I've just put a pink dot on the 5 carbons in this molecule. Chemists refer to the position of the carbon with the terminology 1', 2', 3', 4'. So this is the 1, and I'm going to make a little circle, prime carbon. This is the 2' carbon. This is the 3', 4', 5' carbons, right? That's important because notice that it's the 5' carbon that connects the sugar to the phosphate, and it's the 3' carbon that connects this nucleotide. This is one. I'm gonna draw a little line around it now. Actually, I'm gonna box this up. This is I'm gonna write it here. Nucleotide. Okay. It's the 3' carbon that has the bond with the next phosphate. There's the next phosphate. And then we're going to draw another one of these little sort of right angle bond there. And 1, 2, 3, 4, 5, our next base. I'm going to make this one small because I'm going to make a point about that in a second. 1', 2', 3'. This is the corner that goes to the next phosphate, and so on. I'm just going to make 3 of these nucleotides, okay? And I'll make this one big again. Okay, so there is one side of the double helix molecule. Now, each base, and this is the important part that you're really going to be seeing a lot of when we get back from spring break, each base is paired with another base. So there is there's going to be a, let's see, I have to decide what this is. You guys should know, by the way, that the letters of DNA are A for adenine, T for thymine, C for cytosine, and G for guanine. This is what is referred to as A, T, C, and G. The base pairing is A with T and C with G. So A is always bonded with T, C is always bonded with G, period. That's how it goes. So you can remember this because it spells at Cedar Grove. So I have to decide, let me decide what this is going to be. This is going to be an A. So it's going to be bound to a T. So I'm going to write A in there just as an example. Let's say that's an adenine, right? It really could be anything. This is going to be double hydrogen bonded to a T, which is going to be a small one. I'm going to label in here that these are hydrogen bonds or H bonds, right? So A with T gets a double bond. I made 2 little dotted lines in between. And then we're gonna go to the sugar here, but the important thing to draw here is the other side of this molecule is gonna be flipped. It's gonna look like flipped upside down, right? So you're gonna have to draw this house shape. I'm probably gonna screw this up the first time I do it. You're gonna try to draw this house shape, but upside down. That's good enough. Like that. So there's that nucleotide, right? Now, let's say this is going to be let's say this will be a C. Notice that A's and G's are big. They're called purines. I'm going to leave this out of the notes for now because it's not crucial. But I just want to make a point. A's and G's, this will be a G, are big. C's and T's are small. So again, these are purines and pyrimidines. I'm not going to make you guys worry about it too much, but just to make the point of why I'm making some small and some big. C's and G's are triple hydrogen bonded to each other. So here's a